Okay, we're a little early, but I think everybody's here. So I'm going to hand over to Bill Hill, who I hope won't make quite as big a mess as sharing it as I have.
If you make the slide changes work, I'll be fine. Anyway, thank you very much, Brian. It's my pleasure to chair the second session, and I won't waste a lot of time by yakking in advance. Our first speaker is Sharon Browning from University of Washington. She's actually a New Zealander by origin, as you probably realise when you hear her speak, and she's done some very nice work on long distance identity and such like. So Sharon, is that the problem? The mic's too low. Oh, is that better? Yeah. Pardon? Right. I came that way. Or grew that way. Anyway, the next speaker is Sharon Browning from the University of Washington. Well, there she is. I couldn't see Sharon. Okay.
All right, thank you. Thank you, Bill. It's great to be here.
Sharon.
I've got this one. Can people hear me in the back? I'm seeing the thumbs up. Okay. Yeah, let me know if it becomes a problem. All right. So relatives have correlated values of their traits because of Mendelian inheritance, and that is because of identity by descent, which is the subject of my talk today. So in this picture here, we have a grandmother and two of her grandkids, and we're showing one chromosome pair for this grandmother. So she has a light blue copy and a white copy, and her grandkids have some genetic material inherited from her and some from their other grandparents. And if we line up the chromosomes for those two grandchildren, we see there's a region in the middle where they both share their grandmother's white copy. So that is a segment of identity by descent. So genetic variants in that region that affect the trait will increase the similarity of these two individuals and their trait values over what would be otherwise. Now, in this talk, we're going to concentrate on distant relatives rather than close relatives, such as in that picture. So we're thinking about population samples, such as this group of individuals here. Probably none of them know that they're related to each other, but of course everybody is related at some level. We just have to go back far enough. And so we're going to use identity by descent from those distant relationships to do heritability estimation and mapping of genetic variants that affect the trait. So just to remind you of a few facts about segments of identity by descent. So again, I'm showing you the picture of this little family to illustrate it. There's the grandmother up here. Now I'm showing the intervening generation. These are two of her sons who are half-brothers, and they're the fathers of these two kids. And as we look down, we see that these brothers have this long segment of identity by descent. And then when we look at the kids, their segment of identity by descent is shorter, and that was because of a recombination between the white and the red chromosomes in this meiosis. So that's the general principle. As we have more and more distant relationships, the segments of identity by descent will be smaller due to recombinations. But the rate at which they become smaller is just 1 over the number of meioses. It's not a very fast decay of those segments. So even for very distant relationships, those segments can still be quite long. So say we think about ninth cousins, so 20 meioses, then the expected length would be 5 centimorgans, which in humans is about 5 megabases. So that's pretty long. And those segments, we can detect them even down to 2 centimorgans or smaller. We can detect those in modern genetic data. So that's one fact that we need. The other fact is that, of course, when we look at any point in the genome, the probability of finding identity by descent there goes down as we're looking at more distant relatives. So it goes down very fast. It goes down exponentially in the number of meioses. And so that means once we start looking at more distant relatives, we may not find any identity by descent anywhere in the genome from that relationship. So since that's the case, so if there are segments of identity by descent, they're big, but there might not be any of them. So why do we care about distant relatives at all? Well, the reason is because when we take individuals from our population, they don't just have one distant relationship, like there might be ninth cousins through some great-great-great-grandparent, but they're related in multiple ways through lots of different common ancestors, lots of ways in which they're related. So just, of course, we know that as we look back up, so an individual, 2 parents, 4 grandparents, 8 great-grandparents, and so on going up, you have more and more relatives until it reaches saturation. So once you start looking further back, when you take 2 individuals, there's going to be a lot of shared ancestors, just because you're basically, the ancestors of the individual are basically all the people who were in the population in the past. So that means then that when we add up over all those relationships, we do end up finding identity by descent between individuals in the population. So here's some examples from real populations where we do find this. So some data from the UK and some data from northern Finland. So these are histograms looking at pairs of individuals in the sample. And for each pair, we detect the segments of identity by descent and we add up all their lengths. So that's what's shown along the x-axis is the total amount that we find. And then we have these histograms. And the key point here is that when we look down at 0, we basically don't find any pairs of individuals that share no detectable identity by descent. Okay, now that's what, so these, when I say UK, these are UK Europeans. So there are some caveats. If we take people from opposite ends of the world, then we might not find any detectable identity by descent. Of course, they still have identity by descent, it's just too small for us to find it with our methods. But in these kinds of more homogeneous populations, everybody has these segments of identity by descent. And of course, in northern Finland, there's a lot more identity by descent because it's a smaller size population, had a small founder size, but still the same effect where everybody shares some identity by descent. Okay, so now I just need to go over a couple of technical things that we're going to need before we can get into the heritability and identity by descent mapping. And the first is the way that I'm going to use the word haplotypes in this talk. So here I'm going to use haplotype to refer to one of the 2 chromosome copies of an individual. So I can talk about this haplotype or that haplotype, and at a certain position talk about say the white haplotype. Okay, whereas the word haplotype can be just mean a sequence of alleles, but that's not the meaning I'm using here today. So the important point is that the identity by descent is a property of the haplotypes, the chromosome copies, rather than a property of the individuals, and that's going to be important when we're trying to think about things. Okay, and then for the identity by descent mapping, I'm going to need this concept of transitivity of identity by descent. So it makes sense if we say haplotype A is identical by descent with B and with C, then it should also be that we can say that haplotypes B and C are identical by descent. It's kind of intuitively the way it should be. But in fact, when we do the detection, if we do the detection of identity by descent in a pairwise manner, then actually we don't always find that because the identity by descent between A and B and between A and C might be detectable, but the identity by descent between B and C might not be detectable. Okay, it's still there, it's just we didn't find it. So we just have to do a little post hoc processing to put in those extra bits of identity by descent if they're needed. And once we do that, then we end up with these fully connected groups. So here we've got this ABC group and then this DEFG group. And all pairs of haplotypes within the group are identical by descent with each other. And we can think of that identity by descent group as representing haplotypes that are all descended from one ancestral haplotype. So those haplotypes will all be identical at the position that we're looking at here. And of course, as we move along the genome, the particular identity by descent groups will change. Whenever there's a recombination, haplotypes will move in and out of groups of identity by descent. Okay, so another thing we're going to need, and this is especially for the heritability analysis, is we need to be able to estimate relatedness using the segments of identity by descent. So what we're going to do is we're just going to, for a pair of individuals, we just find all the identity by descent segments, we add up their lengths, and divide by the length of the genome and factors of 2 or 4, depending on what we're calculating, but we get relatedness estimates that way. And I just want to show how well that works. So these are data, simulated data, starting with real data, but then simulating a relationship on top, and different levels of cousins. So in the first column, we've got first and second cousins, third and fourth cousins, then fifth cousins and unrelated. The fifth cousins often don't have any identity by descent. And on the x-axis, we've got the true proportion of identity by descent in the genome, which is something we know because we simulated it. And then on the y-axis, we've got the amount that we estimated. And the top row is using identity by descent segments to estimate those proportions. And the bottom row is using a SNP-based estimate of identity by descent, of the relatedness. So this SNP-based methods, so like basically the usual genetic relatedness matrix that you get when you use SNPs, so based on just allele sharing, those are good, and as we'll see, the identity by descent segment-based approach doesn't necessarily replace it, but for the close relationships, the identity by descent-based approach gives you much more accurate estimate of the realized relatedness of the pair. Right, so and that's because with the SNP-based methods treat the SNPs as independent, but in fact the SNPs are not independent, and considering the IBD segments accounts for that relatedness and so extracts more information from the data. Okay, so now we're ready to start talking about heritability and about IBD mapping. And first I'm going to show you the technical details, and then I'm going to show you results and data. Okay, so we've got a sample of individuals, and Y is the vector of all their phenotypes. We have a matrix of covariates X, and then a relatedness matrix A. Okay, and this is not unique to doing the identity by descent-based approach. Often people will use a SNP-based relatedness matrix for that matrix A, and that's where all this stuff has really been developed. But we can estimate the relatedness as on the previous slide using the segments of identity by descent, and so we get a new relatedness matrix A, and we just plug that in. Okay, so the idea with this model, so we have the effects of the covariates, but then we have these genetic effects, which are random effects, and we also have environmental effects. The environmental effects are assumed to be independent between the individuals. Usually we take out the closest related individuals to achieve that. And then the genetic effects for the individuals are correlated, and the matrix A gives us, the variance covariance matrix is proportional to the matrix A. And then, so if we can estimate these constants sigma g squared and sigma e squared, then we can estimate the heritability. And there are existing programs to use that. Perhaps the best known one is this GCTA program, and that's the one that we're going to use. So basically we could just use the program. All we have to do is calculate our own relatedness matrix A and give that to the software rather than letting it calculate its own one based on the SNPs. So that's heritability. And IBD mapping, so where we're trying to figure out places in the genome that are affecting the trait, is quite similar. The equation, we've just added in this extra term. So we're still allowing for a polygenic effect, and that allows us to account for the relatedness between the individuals. But now we have this term that is just about the position that we're looking at. So we move along the genome testing different positions. We just test one position at a time, and that's our position J. So the variance covariance of G is as before. We have a similar variance covariance matrix for the genetic effects, the random effects that are related to this particular position. But the covariance matrix here is, rather than being for the whole genome, it's just the identity by descent at that position. So for a pair of individuals, they, at a given position, they might share 0 haplotypes identical by descent, or 1, or 2, that's assuming they're not inbred, we can have other possibilities if there's inbreeding. Okay, so that's what the model looks like, and we're testing whether this variance constant is equal to 0 or not. So if it's non-zero, it means there's something going on at this locus in addition to what's happening genome-wide. So we can also reformulate this model, and that can be helpful. So exactly the same model, but we're going to write it a little bit differently. And to do this rewriting, we have to have the transitive identity by descent. That is, we need to have those identity by descent groups that I showed you earlier. So instead of having one genetic random effect per individual and recognizing that those are correlated, instead we can have one random genetic effect per IVD group. That's these gammas here, and those will be uncorrelated. And I'm not showing you the math of how this works, but basically it's because our matrix of identity by descent can be decomposed using the matrix of identity by descent groups. So this matrix Z of identity by descent groups has as its rows the different identity by descent groups, and then as the columns, the individuals. And an individual is a member of 2 identity by descent groups, not necessarily distinct, but we put in a zeros or ones for which IVD groups they have. So the row for the individual is always going to sum up to 2 because they have those 2 IVD groups. Okay, so now we have, we're looking at these random effects that don't have a correlation structure. So that makes things more flexible, presumably can also make the computation a little easier and faster. And when we formulate the model this way, we can use, we have more options in terms of the software that we can use to fit it. And in particular, what the software I'm choosing to use is this software package called SCAT. It's implemented in R. And this software is not designed for identity by descent mapping. This software is designed for doing rare variant gene aggregation tests. The idea being that there are multiple rare variants affecting the trait all on the same gene, and any one of them only has a small effect on the trait, so we don't test them individually. We're just testing as a group, do they have an effect on the trait? And that's the same thing we're wanting to do with these IVD groups. Rather than test whether any particular IVD group has an effect on the trait, we're testing whether as a whole those IVD groups at that position have an effect on the trait. So we can just, once we've got that Z matrix, that matrix of IVD groups, we can just plug it into the software and get the results out. Okay, so we're going to need genome-wide significance levels. So when you think about a GWAS, say when you're doing a genome-wide association study over the whole genome, we know that you have to choose a pretty small p-value threshold. Often people use 5 times 10 to the minus 8. Otherwise, you're just going to have signals all over the place, most of which are just noise. So we're trying to control the family-wise error rate. The results, the p-values are correlated as we move along the genome, and the correlation for identity by descent mapping is higher. It dies off more slowly compared to genome-wide association testing. So we don't have to do as much correction for multiple testing as we would in that case for genome-wide association testing. So, and the way we get these thresholds is to use some kind of simulation. So we could simulate a null trait, or we could just permute the existing trait that we have. And an example of the sort of threshold, you'll see some in the real data as well, is this one from the Wellcome Trust Case Control Consortium data, which was 6 times 10 to the minus 6. So in general, this does depend somewhat from data set to data set, but here we see it's about 2 orders of magnitude of difference. So that's something to keep in mind when you start seeing the p-values. Okay, so before I show you some results, let's just talk about what we expect to see relative to other methods. Okay, so in these pictures, I'm showing you identity by descent groups. So here there's 7 groups, and each group is fully connected, as it should be. And then in each of these circles is a haplotype, and we're looking at a specific marker, a specific genotype location here, and because they're haplotypes, so they just have the one allele, and we're looking at a biallelic locus, so they just carry the major allele or the minor allele. Now, because this group was inherited, represents some ancestral haplotype, that ancestral haplotype was carrying a certain allele, and so unless there's been mutations since then, which will not generally be the case, the group should all be carrying the same allele, just depending on which allele the ancestor was carrying. So this is, on the left here we have an example for common variants. So, a close to 50-50 max of the different major and minor alleles for the different IBD groups. Now here we have a lower frequency variant. So in this pictured example, only one of the IBD groups is carrying that allele. But then there's a difference when we start thinking about the very rarest variants. That is the variants that have very recently arisen. So de novo variants that just occurred in the last generation, or maybe just a few generations back. So in that case, the ancestral haplotype and everyone was just carrying the allele that everybody in the population had, which is the major allele. And then just after that ancestral haplotype, further down, more recent, this mutation occurred, and maybe there's a couple of people in the IBD group might have inherited that. So one or some small number, part of the IBD of one IBD group is carrying this variant. So you can see, in that case, the IBD groups are partially capturing those rarest variants, but not fully capturing them. But in general, these IBD groups are very good at being proxies for variants, and especially, it doesn't matter whether the variant has been genotyped or not. It doesn't matter whether the variant is easily imputed or not. So if we think, for example, about copy number variants, which can be quite hard to genotype, and because they're hard to genotype, we don't have a good reference for them, we can't really impute them. These variants can be picked up by these IBD groups. So we use a set of steps to estimate the identity by descent and get the IBD groups, but then after that, it doesn't matter which variants we had used in our, had genotyped. So when we look at heritability using identity by descent based methods, now it should be picking up the effects of the common variants and the low frequency variants, those ones that correlate with the IBD groups. It'll pick up to some extent some of the rarest variants, but not really the de novo ones. And it doesn't matter whether the variants were genotyped or not, and it doesn't matter what the linkage disequilibrium patterns were. So if you're familiar with the heritability analysis based on STIPs, you have to be a little careful about linkage disequilibrium, and you have to recognize that the variants that weren't genotyped and are not in strong linkage disequilibrium with your genotype variants, their effects are not going to be included in your heritability estimate. So that means we might be able to capture a bit more of the heritability with this type of approach. Okay, then for identity by descent mapping, if the causal variant is genotyped, then we're best to just test it as it is. But if the causal variant is not genotyped and has not been imputed or not imputed well, then the IBD mapping might be able to pick up that signal. And the other thing about the identity by descent mapping is that it's doing an aggregation. So we're testing for a joint effect of all the IBD groups, and that means if different IBD groups represent different causal variants, then those effects will be combined in our test, and that will increase the power. So like with the rare variant gene aggregation type tests, if there's those multiple low frequency variants, then we'll have a better chance of finding them than just the usual SNP-based association where we test one variant at a time. So I'm going to move now to show you some results and some data. So the first data set we're going to look at is the Northern Finland birth cohort. So these are people from up here in the top of Finland, and we've got about 5,000 individuals genotyped on a SNP array and 9 traits, quantitative traits. So before we look at the results, it's interesting to look at the effective size of this population. And this population is a founder population, had a very small effective population size until recently. And that means there's a lot of identity by descent. That makes it really good for these identity by descent based approaches. So in this picture here, I'm showing you effective population size that we estimated using identity by descent. I'm not going to tell you about the method today, but on this plot we have generations before present. So 0 is now 50 generations ago, so maybe about 1,500 years ago. And here's the estimated effective size on a log scale. And you probably can't see it, but there's a line which is the estimate, and then a region, colored region around it, which is our confidence interval. And the Northern Finland birth cohort is this blue line here. And just for comparison, we also have ALS is a cohort that's more Finland-wide, so it has a larger effective population size. So just focusing on the Northern Finland cohort, so we've got this very small effective size until about 15 generations ago, which corresponds roughly to when the area began to be populated and then rapid growth. Now, this is not the effective size of the individuals who were living in Northern Finland 50 generations ago. It's the effective size of the ancestors of the individuals who are now living in Northern Finland. But we've got this bottleneck effect because it was a small number of individuals who populated Northern Finland. So here are our estimates, and I don't want you to worry too much about the individual numbers, but so we've got the traits down here. In this column, we've got the heritability estimate that we get from GCTA if we use it in its normal mode using the SNPs. And then over here, we've got GCTA when we use the segments of identity by descent to construct the relatedness matrix. So there are a couple of things to notice. Now, in each case, we've got the estimate, and then its standard error, and then the little stars help to guide you as to whether it's actually significant or not. And one thing we notice right away is that the standard errors for the regular GCTA are a lot smaller than the standard errors for our IBD-based method. And that's even though we have a lot of identity by descent in this population. It'd be worse if we tried to do this, say, in the UK with a much lower rate of identity by descent. So even though, as we saw, the identity by descent is really good for estimating the very recent relatedness, and in fact, it should be pretty good going back to, say, 30 generations ago or so. I guess there's relatedness, further back than 30 generations that is relevant and informative that the SNPs are picking up, which is giving the smaller standard errors for the SNP-based approach. On the other hand, although we've got these higher standard errors, we potentially could be picking up more of the heritability due to those ungenotyped variants. And I don't have any rigorous demonstration that that's the case here, but if you look, the estimates that are significant do tend to be higher with the IVD-based method, and that's what we'd expect, but we might need a larger sample size to really see that rigorously. So just going back here, so the most significant result that we find is with the LDL cholesterol. And so that's the one that we're going to follow up here with IBD mapping. So our genome-wide significance level is about 10 to the minus 5 here. And the results that we find, we have 2 of them. So one is a PCSK9 on chromosome 1. So this locus was not found during the genome-wide association study of these data, but it is a known locus for LDL cholesterol. And then our second locus is this APO cluster on chromosome 19. This one is also found in the GWAS. It just, well, I guess in these data you don't necessarily need a 5 times 10 to the minus 8 threshold. So, but anyway, it's significant in both analyses. And then in the genome-wide association analysis, there were other loci as well. So IVD mapping definitely doesn't replace genome-wide association analysis, but it can find some additional things, especially when you're set up with a population like this with a small effective size in the recent past. So I'll show you a few results in my last few minutes here from some data from Africa. So these data are unpublished data from Sarah Tishkoff. We've got about 2,000 individuals in genome-wide SNP arrays, and the populations include Cameroon, Ethiopia, Kenya, Tanzania, and Botswana. And we've got 24 traits that are of interest and that are easy to measure in the field. So we've got a number of size-related traits, pigmentation-related traits, and some metabolic traits like cholesterol. So here's our estimates of effective size in these populations. And so there's a bunch of different populations. I should have said that she's gone for the populations that are more like the hunter-gatherers and the unusual populations rather than the more dominant type populations. And mostly the effective sizes of these populations were around 10,000 or maybe 20,000 or so in the past. And then around 10 to 20 generations ago, there's an effective population size reduction, presumably caused by the chaos induced by colonization and so on. And one population in particular had an extremely strong bottleneck. So this is the Hadza from Tanzania. And their effective size dropped to not that much more than 100. So there's a lot of identity by descent in that population. But overall, all these populations have a lot more identity by descent than you'd find in a population like the UK. So these results I'm showing here are very preliminary, and they may change with later analyses, but just to give you an idea of what we find. So we're using SCAT, again, to do this analysis, and here's the genome-wide p-value threshold that we get based on simulating some traits. And because these are preliminary, I'm not going to show you exactly where in the genome these results are, but the results, we found significant results, and for many of these traits, several results. So knee height, arm length, arm circumference, so some size-related traits, but we didn't get significant results for height or weight or BMI. And then some of our metabolic type traits, HDL cholesterol, adiponectin, C-reactive protein, but we didn't find significant results for the pigmentation traits. And one thing, so we haven't done a proper comparison with the genome-wide association study, which her group is doing. We haven't done that yet. But one thing that does seem clear is that the traits that are influenced by a lot of common variants, like the pigmentation and height and so on, those are not really coming up in our IVD mapping, but we're getting a different set of traits. Presumably these traits are being influenced by lower frequency variants. So hopefully we'll find some complementary results to what was found in the GWAS. And that's all. Thank you. Take questions.
Well, thank you very much, Sharon. That's a nice survey of technology which some of us don't know too much about and ought to know more about than we do and that we use. Questions?
This may be obvious to folk who know more about this than I do, but how are you defining your IBD segments? What's the relationship between identity by descent and identity by state? So it seems reasonably obvious to me that the longer the segment of identity, the more likely it is to be IBD. But where do you set the lower boundary?
Yeah, so there's different ways to do it. Basically, what we look for is, so we're looking for these long segments of identity by state, as you say, and then we're looking to see, like we have some statistical model for haplotypes along a chromosome, and we're seeing whether what we're seeing is longer than we would expect to see just from the random background. And then the work that we've done shows that we have pretty good accuracy for the segments we find, we've set a fairly stringent threshold so that once we find them, we're pretty sure that most of that segment is due to inheritance from one recent common ancestor.
There are 2 or 3 other questions down here. I'm not sure. Walter, okay. Oh, he's got the mic's gone behind.
Yeah. Can you say a little bit about the precision or granularity of IBD mapping compared to single SNP mapping?
Yeah, so sure, as we saw, the genome-wide thresholds are lower, and that's because of higher correlation. And so, indeed, the regions definitely are larger. And so something you might want to do is, once you find an IBD mapping signal, you might go look at the association results and see if there's something in there that didn't reach genome-wide association, but might be part of what's driving the signal.
I really had sort of the same question as the first one, because I worry a lot about this definition of haplotype. So haplotype has to be defined by the length and the position, of course. And you say you estimate it based on what the average might be, but that has to take into account linkage disequilibrium, the density of SNPs, whether you allow, I mean, if you happen to have 100 and there's one in the middle that doesn't match, what do you do about that? It's never been clear. And then beyond that, you're just using it, the haplotype, as if it were a single entity without taking any notion of the length of it, because the length of it is going to give you more information if it's longer than if it's shorter.
Yeah, so I guess the length of it tells you how old it is, and maybe if you were looking for
Not necessarily if there's selection involved.
Okay, yeah, so I guess, I don't think about selection very often.
Think about HLA or any of the immune loci.
Yes, sure.
Well, let's have a... You've got to finish, Sharon.
No. Well, I kind of got lost there, I think.
So when you used your method to estimate effective population sizes and say, when you use that part of the method, have you compared it to, say, those coalescent-based methods like MSMC to see how the two compare?
Well, so the coalescent-based methods can only, at this point, work with quite a small number of individuals, and as a result, they can only estimate the effective size further back in time than what we're looking at. So we're looking at the last 100 generations, say, and they're looking at more than several hundred generations ago. So we might see some correspondence between what we're seeing 100 generations ago and what they're seeing 1,000 generations ago, but we can't altogether tie it up. Yeah.
Okay, well, I think we have to finish now. Thanks very much again.
Thank you.
The next speaker is Heather Cordell from Newcastle, who's going to talk to us about regional IBD analysis, linkage analysis and extended pedigrees using genome-wide SNP data.